Good afternoon, everybody. Thank you for taking time to come to our final HMEI Faculty Seminar for the semester. And just as a reminder for those of you here in person, we're very excited that we can be in person. And for today, just remind you that we're going to have masks in the seminar. Please remember to keep them on. So today we have our speaker is Professor Cassie Stoddard from the Department of Ecology and Evolutionary Biology. Cassie is a phenomenal scientist. She is a wonderful communicator in my experience. And she works on what I think is a fascinating set of problems, some of which we'll hear about today involving the eggs. And a few things that I'd like to mention, I have some of them written down so that I can be specific about the actual awards and honors. The one I will start that Cassie received her PhD from the University of Cambridge, correct? And following that, spent some time at Harvard before coming here. And she has a long number of awards and honors, all well-deserved, including in 2020 being elected a fellow of the American Ornithological Society. So that is quite an achievement and an honor. And so one of the parts that I really like in the bio and actually in the way Cassie works is that she has been, what is it called? It's a curator or a curator, curatorial duties, and as a research fellow at a few museums, including the one at Yale. But for me, the one that really stands out is the American Museum of Natural History. I think many of us that have had the experience to go there know what a wonderful place it is. And so I have a massive jealousy that you've had to be involved with there. And Cassie uses a variety of methods to understand birds and the relationship to the environment. One of the things that I'm not sure we're going to hear about today, but I'm so excited to hear about, maybe not today and another day, is what are these glasses that allow us to see the world like a bird? Is that correct? Really sort of innovative ways to get us to understand birds and the relationship to us. So without any more, I would like to welcome Cassie to give us a wonderful talk.
Well, Gabe, thank you so much for that very kind introduction. I am really delighted to have the chance today to talk about my favorite topic, colorful birds and their spectacular eggs. And I just want to start by saying how fortunate I am to work with a really wonderful team of students and postdocs here at Princeton and at our main field site at the Rocky Mountain Biological Lab. Several of the photos in this talk were actually taken by Julian and by Kojo, two undergrads who joined us in the field last summer. As you can probably guess from our binoculars here, we study birds. So why birds? Well, as evolutionary biologists, we are really interested in the historical processes that generate diversity. So when we look at a scene like this, we ask, why do animals and plants come in so many different varieties? And how do they get this way? And birds are a fantastic system for thinking about these questions for a few reasons. The first is that birds are the most diverse terrestrial vertebrates. There are over 10,000 species and counting. They live on every continent, and they inhabit a huge range of ecological niches. Birds are also the most colorful land vertebrates. Their visual signals, you should see here in full display, are extremely diverse. Birds can make their colors with pigments. They can also make their colors with nanoscale structures in the feathers themselves. And they often produce these wild and fabulous visual displays. Sometimes the colors are combined with elaborate dance moves or acoustic components to produce some of the stunning displays you see here. In addition, birds have excellent color vision. So not only do they produce color for attracting mates or for providing camouflage, birds also can see way more colors than we can. Birds have 4 color cone types in their eyes. We humans just have 3 color cone types in their eyes. So that means that birds are probably experiencing an even more colorful world than we imagine. So here's a very iconic, colorful bird. This is the painted bunting. And I was in college when I first used a spectrophotometer to measure the greenback of this painted bunting. And I discovered that this greenback isn't actually green. It is UV green. So we humans can detect light between 400 and 700 nanometers. Birds can see all of these colors, plus colors in the ultraviolet range between 300 and 400 nanometers of light. So to a bird, this UV greenback probably looks even more colorful to us than we can imagine. Here are some photos that we took of a rainbow lorikeet using a regular camera and a UV-sensitive camera. So you can see here that the blue feathers on the head are actually UV blue. They're reflecting a lot of ultraviolet light. And here's an even more extreme example. To us, this superb fairy wren appears to have a solid, uniform, bluish-black throat. But when we take an image of this bird using a UV-sensitive camera, we can see this hidden UV patch is sort of thrown into light. So there's much more here than meets the eye. Another reason we study birds is that their eggs are extremely variable in their color and their pattern and their shape and their structure. And this is actually pretty unusual. Most vertebrates lay eggs that are unpigmented and they're relatively simple in terms of their shape and their structure. Finally, we study birds because they're very good bioindicators. Birds are very sensitive to environmental stressors, so they often provide a picture of an ecosystem's health. And birds are not doing particularly well. About 1.6% of bird species have gone extinct since the year 1500. And here in North America, in the last 50 years alone, 30% of the bird population has disappeared. And that's a whopping 3 billion birds. So there are many reasons to study birds. They're really colorful, they lay impressive eggs, they're ecologically important. In my group, we're specifically interested in the functions and mechanisms of avian color vision, and the evolution and engineering of bird eggs. And these topics really do invite an interdisciplinary perspective. So we're often using tools from optics and physics and math and computer science and engineering. Our work is based in the lab, in the field, and as Gabe mentioned, in the collections of natural history museums. These collections are really important for our work. And increasingly, our research is intersecting with questions about biodiversity and climate change. I won't have time to go into detail about all of the projects happening in the lab right now, but I did want to provide a sort of bird's eye view. So in terms of the functions of bird color, Monica, a PhD student in the lab, is working on woodpecker plumage mimicry and camouflage. Jerome is exploring plumage evolution and speciation in parrots. And Audrey is studying dynamic courtship displays in hummingbirds. In terms of the mechanisms of bird color, Clara is investigating the evolution of iridescent feather nanostructures, and Rosalind is working on avian color genetics and feather microstructure. On the color vision front, Ben, a postdoc in the lab, is working on hyperspectral imaging and hummingbird color vision. And the team working on eggs consists of David, who's studying the function, structure, and evolution of eggshells, Delina, who's working on the genetics and evolution of paleonate eggs. The paleonates are ancient birds like ostriches and emus. And Samudu, who is investigating the eggs and nests of many birds in the context of different climatic niches. Today, I want to share 3 brief stories. The first is about how hummingbirds see the world. The second is about hummingbird courtship and foraging behavior. And the third is about the evolution and engineering of eggs. So as a spoiler alert, we do not have goggles or glasses like Gabe mentioned to see the world like a bird, but we have been training hummingbirds to show us through their behavior what colors they can see. And that story starts with the broad-tailed hummingbird. This is a broad-tailed hummingbird. We believe that hummingbirds, like most other diurnal birds, are tetrachromatic. That means that they have 4 color cone types in their retinas. These are sensitive to ultraviolet, short, medium, and long wavelengths of light. In other words, birds have a UV sensitive cone, a blue sensitive cone, a green sensitive cone, and a red sensitive cone. By contrast, most mammals are dichromatic with just 2 color cone types in their eyes. These are usually sensitive to short and long wavelengths. Humans are trichromatic. We have 3 color cone types, our blue cones, our green cones, and our red cones. Bees are also trichromatic, but they have a UV sensitive cone instead of a red sensitive cone. So what all this means is that you can have a marmot, a human, a bee, and a bird all living in the same meadow, but they'll be seeing this meadow in very different ways. So birds are tetrachromatic, but what does that really mean? Well, we think that color vision has to do with the way that the different cone types are interacting. So it's not just that birds can see all of our human colors with a little UV tacked on. In theory, birds should be able to see a whole range of combination colors like UV red and UV green. And we think this will give birds an extra dimension of color perception. So in humans, all of the colors that we can see can be represented in this simple triangular color space. And each vertex represents one of our cone types, our shortwave sensitive blue cone type, our medium wave sensitive green cone type, and our long wave sensitive red cone type. So when we see white, all 3 of our cone types are equally stimulated. When we see turquoise, that's when our blue and green cones, but not our red cones, are stimulated. Because birds have a 4th color cone type, we can represent their colors in this tetrahedral color space. And we currently assume that birds can see colors throughout this entire space. And that's a very convenient assumption because it allows us to measure bird colors in a way that accounts for their visual system. So we can measure the reflectance properties of this painted bunting's blue crown or the reflectance properties of its UV green back. And we can combine this with information about the sensitivities of the bird's 4 color cones. That allows us to estimate how a bird's eye would be stimulated. That enables us to map plumage colors in this color space. But we still have a pretty limited understanding of how birds are perceiving colors throughout this space. In fact, carefully controlled color vision experiments in the wild are very rare. Most work on color vision has been done in the lab on a few model species like chickens and zebra finches. And this makes sense because in a lab, you do have control over lighting, over weather, and over the birds that you're studying. But the problem is that these studies in the lab have not been validated in the field, out there in the real world where birds are living and behaving. So we lack systems for investigating color vision in wild animals. To address this, we took inspiration from some past work on hummingbirds and a lot of work on bees, and we turned our attention to the broad-tailed hummingbirds. And we study these wonderful birds in the Colorado Rockies at the Rocky Mountain Biological Laboratory. So this field site is at about 3,000 meters elevation. The broad-tailed hummingbirds spend their summer breeding here after migrating north from Mexico. So they'll spend a few months here in the Rockies, and that's when they are finding a mate, they are building a nest, and they are rearing their young. Now, most of the year, the field site is completely covered in snow. This is what it looks like. But by the time we arrive in early June, the snow has usually melted, and it looks like this. And I've been working with my colleagues, including Ben Hogan, David Inouye, and Harold Eister, who you can see here, and also Ed Susi and Dylan Morris. And we together have been training a population of wild hummingbirds to participate in color discrimination tests. I say training here, but really the birds require very little training because they'll visit feeders regularly. And we've been capitalizing on this by hanging up feeders to attract the birds. And once we've taught the birds that this is a very good site to visit for getting food, we take down the feeders and we replace them with our own experimental setup. We designed these special bird vision light tubes. These are LED light tubes. We call them the tetracolor tubes because they are capable of producing a whole range of bird visible colors like UV green. And we've been performing experiments that work like this. So in these experiments, one color is rewarded with sugar water. The other has just water. And after a set time, we swap the positions of these tripods so the birds can't simply use spatial cues to select the rewarded color. We then record whether the birds choose the correct rewarded color first when they visit the experiment, which they're doing several times over the course of the day. So after several hours, we can actually detect at the level of the population whether the birds are able to distinguish between the two colors. And in this case, the birds could easily discriminate between red and green. And they could do this when we rewarded red. And they could also do it when we rewarded green. So we did these experiments back to back, and they can easily learn and unlearn and relearn these color associations. We also match the colors for brightness, so they're equally bright. We also perform control experiments, which show that the birds cannot distinguish between identical colors when one color is rewarded and the other is not. And that might be the case if the birds were using some other cue. Maybe they're copying each other and using social cues. Maybe they're using smell. So these control experiments are important and suggest that the color is really the key variable of interest here. So here's a brief clip showing the birds participating in an experiment. This was a harder test where the birds had to distinguish between a red light and a UV red light. And these experiments typically run for several hours as the hummingbirds are coming and going as they please. And over time, we can detect whether the birds in the population have learned to distinguish the rewarded color from the unrewarded color. Now, the experiments that I showed you in the cartoon, red versus green and green versus red, these are very simple experiments, and the hummingbirds have no trouble distinguishing a green light from a red light. But using this same paradigm, we've been performing more complex color discrimination tasks. So one of the big questions we've been asking is, can hummingbirds actually see all the colors throughout this color space? This is a fundamental assumption of the color vision models that we use in birds and other tetrachromats, and it relies on the idea that birds can see non-spectral colors. But this has not been tested systematically. So what are the non-spectral colors? Well, unlike the spectral colors, which you see here on these three colored lines, the non-spectral colors are not in the rainbow, and they fall on the three other axes. The axes here shown with dashed lines. So UV red is not a color in the rainbow. Neither is UV green, and in fact, purple is not in the rainbow either. UV green colors will predominantly stimulate the UV and the green cones. UV red colors will predominantly stimulate the UV and the red cones. And then purple colors will predominantly stimulate the blue and the red cones. These are colors that stimulate pairs of cones that are widely separated by big bands in the spectrum. So these are unusual combination colors, and we don't know much about how and whether birds can distinguish these non-spectral colors. Evidence from lab experiments suggests that the spectral colors are highly visible to the birds, but what about these non-spectral colors? And I think one of the reasons for our having ignored non-spectral colors in the past is that we humans have only one major type of non-spectral color, and that's purple, which arises when our red cones and our blue cones are stimulated. Purple, as I mentioned, is not in the rainbow. It is a special color that arises because we have trichromatic color perception. As I mentioned, birds have many additional possible non-spectral colors, UV green, UV red, and purple being the main types. So we've now done a series of experiments along these color axes in color space to demonstrate that hummingbirds really can discriminate these non-spectral colors. So in other words, the hummingbirds can distinguish UV red from pure red, and they can distinguish UV red from pure UV. And they can even distinguish between two different mixtures of UV red light when one has a little more UV and the other has a little more red. So this suggests that hummingbirds really do see a huge range of these combination non-spectral colors that we humans can only imagine. And this is probably true not just in hummingbirds, but in birds and other tetrachromats more generally. These results provide strong support for this tetrahedral color space model. In addition, we wanted to see if these non-spectral colors are prevalent in the natural habitats of these birds. So we measured thousands of plumage colors and plant colors. Here are some reflectance spectra showing these UV red and UV green and UV and purple colors in plumage and in plants. And we estimate that these colors really are much more common for birds than they are to us. When we analyzed 1,000 plumage colors, we estimated that about a third of these would appear non-spectral to the birds. Now, I don't want to give the impression that non-spectral colors are somehow special for these birds. It's not like these colors are glowing in their environment or anything like this. This is just a consequence of birds having a tetrachromatic color system. This is a part of that color vision system that many of these colors will be non-spectral. The take-home message here is that these colors really do exist in nature, and birds can and they do perceive them in the wild. So why does this matter, particularly from an environmental perspective? Well, I think it's clear that we still lack a detailed understanding of avian color vision. And this is a problem because hummingbirds rely on color to find and remember flowers. It's a big part of their sensory world. We know that hummingbirds' landscapes are quickly changing from climate change and land use change and wildfires. And if we really want to protect and preserve hummingbirds' habitats, we have to understand how they perceive it, not how we perceive it. And this idea is really gaining some traction in our community. This is the idea that conservation biology is really enriched by these sensory ecology perspectives. So with all of that in mind, I want to turn now to the colorful lives of hummingbirds, particularly with respect to courtship and foraging behavior. Specifically, we've been asking, what role does color play in courtship displays? And what flowers do hummingbirds visit? And how does this change across the summer and across different years? Well, one thing we remember seeing for the first time when we went out to the field and saw these broad-tailed hummingbirds is that the males perform really impressive courtship dives in which they fly about 30 meters up into the air, and then they do this spectacular horseshoe-shaped dive over the head of a female, and then they do it repeatedly. So they're swinging back and forth like a pendulum. Just as they go over the head of the female, they're snapping their tail feathers together to create this loud buzz, and they're showing off their iridescent throat feathers. So with the postdoc in the lab, Ben Hogan, we wanted to know how is color interacting with motion and speed and sound in this dive display? How does the female perceive these different elements, and are they highly synchronized? Historically, it's been relatively easy to study visual signals in a static way, but real animals move. And this dive of the hummingbird represents a signal that is complex and dynamic in both space and time. And we really wanted to account for that in this study. So in the field, we recorded videos of dives, and we used tracking software to map the path of the male's dives. So here's a male. It performed 2 sequential dives. Now, the male is too small for you to see here, but we tracked him frame by frame, and that's what you can see with these red dots. From the tracked dives, we could then calculate some flight performance statistics. For example, males on average are going 25 meters per second at the base of this dive. That is really fast. We also analyzed the sounds that the male is making in the dive. So here are sonograms from 5 different dives, and they all show the same general pattern. So as the male is ascending, he's making a trill produced with the wings. At the base of the dive, he's no longer using his wings, he's using his tail feathers to snap together and make this buzz. And in the sonograms, you can actually detect this buzz. And using this acoustic information, we could start to map these sounds back onto the trajectory of the dives to see how everything would time up. The final component was color. We used multi-angle UV and human visible photography to capture iridescence of the plumage of museum specimens. We designed a 3D printed stage for hummingbirds to which we attached the specimen, and then we rotated it and imaged it at fixed angles. And here's a video that really highlights the striking degree of that color change. And we did this work at the American Museum of Natural History in New York. I agree with Gabe, this is a remarkable museum. And behind closed doors, not on view to the public, is one of the largest collections of bird specimens in the world. And we've been very fortunate to work there. This is Paul Sweet. He's the collections manager in ornithology, and he's been a really major facilitator of this work. We then used a model of bird color vision to estimate how the female's color cones would be stimulated by the male's iridescent throat. And this showed us that as the male dives toward the female, he'll first appear very red, but as he reaches the bottom of the dive, his throat will appear to change quickly from red to a blackish green. So putting this all together, for each dive, we could investigate the timing of these three key features. Here's a simulation for one dive, and it really shows that a male is delivering this sensory explosion right at the base of the dive. So here the perceived color will appear to change from red to black. He's making his tail-generated buzz, and he's also reaching his top speed. When we analyzed all of the dives together, we realized that this was a general feature. Males are reaching their top speed at almost exactly the same time that they're snapping those tail feathers and showing off their iridescent throats. So in other words, all of the action is happening at once in what we estimate to be a 300 millisecond window. So if you blink your eyes, that's 300 milliseconds. So we now have a better understanding of how these remarkable displays are unfolding and how very tightly synchronized they can be. But how do males get the energy to perform these dives? Well, of course, they are tanking up on nectar from a variety of wildflowers. And at our field site, every week in the summer brings a new wave of wildflowers. So here are just a few of the beautiful wildflower species that we see out in the Colorado Rockies. The hummingbirds, of course, rely on pollinators like hummingbirds and bees, and hummingbirds rely on the nectar rewards of the flowers as a major food source. But these mutualisms are very fragile. They require overlap in the timing of the flower bloom and the timing of pollinator activity. If climate change accelerates flower bloom times, then there could be a phenological mismatch. In other words, hummingbirds, remember they're migrating from Mexico, may no longer be able to coincide the timing of their breeding behavior with the peak bloom of wildflowers. Thanks to long-term data on flower bloom times at the Rocky Mountain Biological Lab, we actually know that some of the hummingbird's favorite flowers, like this blue delphinium and this yellow erythronium, are blooming earlier than they did 50 years ago. We estimate that this is changing the visual landscape experienced by hummingbirds. So we can actually go back in time and look at a particular week in 1988, and we can see that in this week, the hummingbirds were mostly seeing yellow erythronium flowers. But during the exact same week in 2012, the hummingbirds mostly saw blue delphinium flowers. Now, while we understand quite a bit about what the flowers are doing, we actually don't know much about what the hummingbirds are doing. We lack good quantitative data on hummingbird foraging patterns. So we want to know what flowers are the hummingbirds actually visiting and with what frequency. So to answer this, we've been using these time-lapse cameras to catch hummingbirds in the act of foraging. We set up these cameras at different wildflowers. We record for 3 days, and then we move the cameras to new flowers. We've been focusing on about 20 species that we know to be visited by hummingbirds. And once we download all of this footage, we use a machine learning algorithm to return to us the images that contain hummingbirds. So I'm going to show you just a highlights reel here. And from these data, we can learn which flower species are visiting, how frequently flower species are being visited, and when the hummingbirds are visiting, what time of the day, how is this changing across the season. So far, we have obtained about 40,000 flower hours over 4 summers. We've detected more than 2,000 different hummingbird visits, and that's a rate of about 0.5 visits per hour of observation. So you would have to watch a flower for about 20 hours to get 1 visit. So this kind of technology, this kind of approach is really giving us an unprecedented glimpse into the foraging lives of these birds. It's showing us not just what the hummingbird's favorite flower species are, it's also allowing us to build a very detailed record of the flower species visited by hummingbirds throughout the summer, including species that the hummingbirds don't visit very often. So here's a map of how hummingbird foraging patterns are changing for different floral species as the summer unfolds, but we're also able to look at differences across years. For example, 2018 was a very hot, very dry summer. 2019 was a very cold, very wet summer. And the snowmelt date in 2019 was 39 days later than it was in 2018. As we suspected, most flowers bloomed earlier in 2018 than in 2019, and we predicted that 2018 would be a very rough year for the birds. That was the hot, dry summer with conditions that we will expect as the climate warms. And in a way, this was true. 2018 was so hot and snowmelt was so advanced that some of the early season flowers, like erythronium, had already wilted by the time the hummingbirds had arrived. In general, we did see that the hummingbirds were visiting flowers earlier in 2018 than in 2019. They were mirroring the flowers' bloom times. But surprisingly, we saw that 2019, this cold, wet year, was also very challenging for the birds. So it turned out that because the snowmelt was so late in this year, very few of the hummingbirds' preferred flowers were available in the early part of the season. And we think this resulted in a more sort of frantic and diverse set of interactions with uncommon, low rewarding, low nectar plants later in the season as the hummingbirds were struggling to compensate to meet their nectar demands after weeks of an impoverished floral supply. So the bad news is that we're already seeing the effects of climate change at our field site, and it's leading to the kinds of phenological mismatches that could be challenging for hummingbirds and flowers. In addition, climate change is likely contributing to these more extreme swings from year to year, which is why we observe this very cold, wet year with delayed snowmelt, which also caused problems. So I think the broad-tailed hummingbirds may suffer this sort of double whammy where they're going to experience negative effects of both early and late flowering. But there is some hope. The hummingbirds do appear to be quite flexible with respect to the plants they visit, and it looks like these plant pollinator networks can be rewired. So it's possible that they will be robust enough to respond to these changes and switch up their food resources. It does seem clear, no matter how you look at it, that the landscape is changing. Just last year, U.S. Fish and Wildlife added broad-tailed hummingbirds to a list of birds of conservation concern. They cited declining populations. And so these declines may have been influenced by just the kinds of shifts and mismatches that we're already seeing at our field site. On a happy note, this project, Hummer Flower Watch, and I see some of the undergrads here in the audience, would not have been possible without a super group of Princeton undergrads. They come out every summer to collect and analyze these data, and their energy, especially during the pandemic, when we weren't able to do the work as we planned, was really just amazing. So our work in the field has explored how hummingbirds perceive and use color, but back in the lab, we're trying to come up with new ways of measuring color. For example, we've developed a bird vision hyperspectral camera that allows us to obtain really high resolution color information for every pixel in an image of a specimen. And this project is being led by Ben Hogan. We're also building and animating 3D models of museum specimens, each of which contains UV information that is relevant to birds. And this project is being led by Audrey Miller. Finally, we're taking a close look at the feather nanostructures responsible for iridescent feathers, like the ones you see on hummingbirds. And in this project, Clara Nordin was exploring how different bird lineages evolved very diverse feather nanostructures, like you see here in many iridescent feathers. And we discovered that the different nanostructures in these iridescent feathers, despite coming in a wide variety of shapes, are all equally good at producing these vivid, saturated, bright iridescent colors. So we think birds actually have a very flexible nanostructure toolkit, and that there's more than one way to make a really dazzling iridescent feather. So to recap, hummingbirds could be an excellent system for investigating color vision in the wild. They can see way more colors than we can. They rely on color in courtship and foraging, and hummingbird foraging patterns will be affected by climate change. Finally, I just want to say a few words about bird eggs. So we found that bird eggs can be an excellent model system for exploring the diversity of color and pattern and shape and structure in nature. We've worked on egg mimicry by brood parasites. We've worked on egg camouflage. We've studied eggs as potential reproductive signals to mates. And now we've gotten really interested in egg shape and structure. It turns out that bird eggs come in a tremendous variety of egg shapes, and that got me very interested in the eggshell's fine scale structure. So do eggs vary in terms of their structural and biomechanical properties too? So my colleagues and I have been studying the eggshell structure of the beautiful green eggs of the emu. This is a large flightless bird from Australia. And in emus, it's actually the male that incubates the eggs. He will lose up to 1/3 of his body weight during the incubation period. So this suggests that egg breakage is a real risk for these birds. And my collaborators and I want to understand how emu eggshells actually work. Eggshell is one of the, in my view, most interesting natural materials because it is actually built to break. If you compare eggshell to other biominerals like your teeth or your bones, these are not supposed to break. But eggshell is simultaneously strong and fragile. It's also one of the fastest forming biominerals in nature. So if you imagine a chicken laying an egg every day, you're going from 0 to eggshell in under 24 hours. And that's very different from the rate of calcification, say, in a mollusk shell. So we really wanted to know, how does the eggshell fulfill these competing biomechanical roles? How is it preventing premature fracture, but ultimately allowing a chick to hatch? So we've been taking a closer look at these emu eggshells. Here's an emu eggshell in cross-section, and this allows us to really see the variation in fine-scale structure as you move from the inside of the egg with these inner membranes to the outermost part of the eggshell. Here's a video that's showing the really detailed internal structure of the different eggshell layers. This is followed by a reconstruction of the pores and the vesicles that are functioning in gas diffusion inside this shell. And understanding the details of this internal structure have been really important for assessing the biomechanical function, the unique biomechanical function of this shell. So to understand how the eggshell really breaks, we've been performing a series of biomechanical tests. We've been finding that in the outer region of the shell, cracks are deflected sideways. This prevents them from breaking through the shell, as you might expect if they're adaptations for preventing premature breakage. But when we look at the inner part of the shell, we find that the cracks are actually easy to propagate vertically, particularly at the boundaries of these inner crystals. So this might help initiate cracking when the chick is hatching. So what we're finding here through these analyses is that there are some built-in adaptations in the egg that make it both structurally robust and also internally weak. The egg won't shatter too soon, but it will allow a chick to hatch. And so this is just kind of a preview of what we're working on, but it's giving us a richer picture of eggshell biomechanics. Okay, so now why do eggshell biomechanics matter if you're interested in preserving the environment? Well, we know that DDT and DDE, these pesticides, caused eggshells to thin in the 1950s and 1960s. This nearly wiped out some bird species, and in fact, some raptors still have not recovered from these effects and won't lay normal eggshells for another 10 years at least. In addition, a study that came out earlier this spring showed that climate change is causing many birds to lay their eggs up to a month earlier. If birds are laying earlier, but they don't have the same access to insects in their diet, they may not be able to get the kind of calcium they need to make shells of a normal thickness and size. So I think understanding fundamentally how eggs break, the biomechanics of eggshell, could be really important for assessing the impacts of pesticides and climate change on egg viability. Now finally, to end here on a happy note, it could be that eggshells are part of the solution. So there's some studies that show that eggshell actually has some carbon capture properties. It's been shown to absorb a lot of carbon dioxide during hydrogen fuel production. So maybe they can be a part of a clean energy solution. So just to wrap up, birds inhabit a colorful world, but we are colorblind by comparison. So we really do have to shed our human goggles if we hope to preserve and protect birds and their habitats. Hummingbirds, of course, are vital pollinators, but they're facing challenges in light of climate change, and we're already seeing evidence of this at our field site. And finally, eggshells are sensitive to environmental stressors like pesticides. Understanding how they're built and how they break is going to be really important. And I think, one thing that I've learned in all of this is that we can use what we're learning to make life better for birds. We're really happy to be working with the Princeton Birding Society, and I see some representatives here, to help minimize bird collisions with glass buildings on campus. This is an area where having a sensory perspective, and particularly a perspective on bird vision, could be really helpful. So with that, I'll say thank you first and foremost to HMEI, which has supported many of these projects and many of these students who've gone out with us to the Rocky Mountain Biological Lab, and of course to the many colleagues and groups that have made all of this possible. So, thank you very much.
Okay. So thank you, Cassie. So we have time now for questions. I see there's some coming in from Zoom, but Reed.
Sure. I need to be loud. Right here. Thank you for a really super awesome talk. That was really great. I was very interested in your work at Rumble on basically the snowmelt timing and different basically wildflower cycles. I'm really curious too about your thoughts on John Hart's work there, the 30+ year warming experiment that shows land cover change and some succession. I'm curious about what that might mean for nectar availability for hummingbirds. Thank you very much.
Yeah, thanks for that question. It's been really a delight to talk to John about some of his work. We've been fortunate to bump into him there. I think that one thing that's coming out of both the work on the flower phenology, but also the succession work, is that there are general patterns, but there are also deviations from those patterns. So whereas we might see, with respect to the wildflower blooms, that many of the flower species are blooming earlier, that's not true for all flower species. And there are some parallels with the succession work as well. So you can see that there are sort of general themes that dictate what we think will happen in the future that give us some predictive power, but there are a lot of exceptions to those rules, which make the situation both interesting and complex. And I think one missing piece of the puzzle for us is what the hummingbirds are actually doing. How are they going to adapt? So initially, I think we went in thinking this was all doom and gloom for these birds, and truly 2018 and 2019 were two extremes of bad situation for these birds. But work by some of our ecologist colleagues there has shown that some of these pollinator plant networks can be rewired even over short time scales, so that the key ingredients for the pollinators getting what they need and the plants getting what they need are actually in place. So I guess I'd summarize by saying that we're still learning a lot. We need the perspectives of successional change. We need the pollinator side of things. We definitely need the plant phenology side of things. And what I look forward to is sort of seeing how this allows us to come up with more accurate models and predictions. It's one of the joys of working at Rumble is that we have inherited all of this wisdom from biologists who collected these data for decades, not knowing how useful it would be to us now.
Okay, so I'm going to take a couple questions here from the chat. First, there's one, I'll give two, and they're not entirely related, but they are. So the first question is, how do birds smell, and how do they know where to come back to for food after migrating? And then another question is, given that female hummingbirds and birds in general are dull in color compared to males, how do you think males actually see the colors of the female feathers? Are they different from what we see?
Okay, great. Those are both wonderful questions. So first, thinking about bird smell. Well, it's an oft-repeated myth that birds don't have good smell. Birds actually can smell, and this has been demonstrated in a number of species. It's most sort of notable in sort of pelagic birds like albatrosses, for example, but there are studies that have come out in the last year showing that hummingbirds can smell too. So I think we're learning a lot more about hummingbird smell. As for how hummingbirds remember where to come back each year, this is one of the sort of most exciting and bewildering questions, I think, in all of bird biology, is how migration works, how memory year after year is actually working, what cues birds are using to find sources that they've visited previously again and again. I can say that some of the hummingbirds that we banned at our field site, at one small part of Rumble, are many times birds that were banded there in previous years. So these birds have a unique identifier band, and we know that this bird received this band on this particular day going back in time. So we are seeing a large fraction of the hummingbirds at our field site as repeat customers. They're coming through to the exact same place. How they do this, both how they navigate, how they decide where they're going to breed, these are questions that are still open, but it's something that we're really interested in. In terms of how the females are perceived, in our group we talk a lot about how we all tend to focus on studying the male and their dazzling dive displays and their spectacular colors, but we really do need to understand the female perspective as well as how males are perceiving these females. The female hummingbirds tend to be white and green, sometimes with some dullish browns. I don't think that we've seen any hidden UV patches in hummingbirds yet, but for sure these greens and whites have a UV component that will look different to birds than they will to us.
Okay. We have a question.
Yeah, I have a question.
So let's wait for the microphone because we have people in Zoom land.
I'll ask a question, but I don't know if it's worthwhile. So I was just thinking about the differences between human perception and bird perception. And one of the things that happens for us is that like our perceptual world is it's like wrought with conceptual content. So like I don't just see the brown lectern, I see it as a lectern, right? And so I was thinking about running the experiments were based on color and thinking about like shape and shape discrimination. And if what they're doing is seeing things like the different outlines of flower shapes as like perceiving it not just as a red blotch, but as a red blotch that's food, oriented or something. And just thinking about like what versions of different kinds of, it's not conceptual, but like other perceptual changes that are not just color-based, that aren't smell or whatever. And so I was thinking of shape. Is there anything like that's going to potentially lead to discriminations on the basis of what I would call in human perception conceptual content? But
no, those are,
sense?
yes. Those are really interesting set of questions there. So I think actually the best work on the questions that you're describing has been done in pigeons. So there are some really cool pigeon cognition labs that have asked questions about shape invariance, for example. Will a pigeon be able to recognize a picture of a hamburger when it's like flipped on its side versus not? And pigeons actually do demonstrate quite an impressive degree of that kind of shape invariance, which could extend to some kind of conceptual learning. I don't know that we have done that level of kind of psychophysical test about these concepts in other wild birds for obvious reasons, but I don't doubt that when a hummingbird is looking at a flower, it's combining its understanding of that flower's shape, its color. You've all seen the pictures of sort of hidden UV patterns that exist on flowers and building up a percept of this as a food source. One question that we've been thinking a lot about in my group is once it has that picture in its mind of what is going to give it food in the first week of June when it arrives at Rumble, how flexible is that picture? Is it going to generalize to other flowers that are also red? Is it going to generalize to other flowers that also have these patterns? So one project that Darcy, who's in the back, is going to be working on this summer, Darcy is an undergrad who will be joining us at Rumble, is building up a library of images in the UV and in human visible wavelengths of all of the wildflowers that birds and bees are visiting to see if we can learn more about some of those general themes that make a flower desirable to a hummingbird.
Hey, we have another question.
Excellent talk. That was really exciting. So maybe you addressed in the first 10 minutes when I came in late. What determines, so one thing that was striking was how infrequently birds go to a particular flower. You know, you said that it takes them about, say, 20 hours before it sees another visitor. Is there anything in the coloration of the flower after a bird has been there that signals that, you know, that the nectar is already exhausted? Or why do they not keep trying each flower that they go to and do this in a sort of a random order? Or is there some signaling going on from the flower as well based on what's being fed?
That's an intriguing question. I think the short answer is we still don't know, but we think that there is some potential there for hummingbirds and bees to pay attention to color changes that happen after visiting so that they don't go and tank up again. I had an undergrad named Cole Morokovich who got really interested in that idea, and he actually grew some delphinium, the blue flower plants, in a greenhouse and then took them out and did a cafeteria experiment. So he sat and watched and waited for hummingbirds and bees to visit. And he hypothesized that there were actually different colors on the plant itself that would indicate whether those flowers had more or less nectar. And this has to do with the development of the delphinium plant. It has female plants and male plants, flowers, all on this, and they do vary a bit in color. Cole did not find any evidence that the hummingbirds were particularly picky in which of the flowers on the plant they got to. They just kind of shopped around and took what they could get. But the bees were more picky. And so maybe there are some differences in how birds and bees and other pollinators are responding to these potential color changes. Another thing is that hummingbirds have different kinds of behavior in how they visit wildflowers. Some hummingbirds are trap liners and they'll go back to the same flowers again and again, day after day after day. Others might go to a patch and sort of exploit that patch generally before going to another patch. So in a perfect world, we'd ultimately be able to use some of these data to learn more about individual hummingbirds and their individual foraging patterns. At the moment, we just know that this is a flower and it was or it wasn't visited by a hummingbird, but there's a richness to the data that we could tap into down the line. Thank you for that question.
Okay, I'm going to take group 2 questions from online. One is there a particular color flower that has more nutrition from hummingbirds? And then Hannah Wang has, I think, the most important question, which is, first, thanks you for an amazing talk, but she was wondering what your favorite bird is. And there's obviously a right answer to that question, but I won't tell you what it is yet.
Okay, good. We'll come back to that. So it's actually very interesting. For a long time, the typical hummingbird flower was thought to be a red flower with a tubular flower shape. Those were supposed to be perfect for hummingbirds. Hummingbirds have co-evolved with these flowers and can get their bills deep into these red tubular flowers that are not visited by insects. So these typical hummingbird flowers often do have higher nectar rewards than some of the other flowers, but we have colleagues at Rumble, Nick Wasser and Mary Price, who did a really interesting study in which they showed that hummingbirds actually often visit atypical flowers that may or may not have reduced nectar content and still get enough for their energetic demands. And so I think we're actually revisiting our ideas about what a typical hummingbird flower actually is. But if you open any book, you will read a hummingbird flower is red and it's tubular. And actually what we're finding, even with our own work, is that these hummingbirds will exploit a vast range of resources, even if they do have preferred flowers. So the next question, I think, is what is my favorite bird? And my lab has heard me say this many times. It's actually not the broad-tailed hummingbird. It is the painted bunting that I showed you with its spectacular UV green back. That is a bird I dreamed of seeing ever since I sort of saw it in a field guide. And I remember the day that I first saw one in Florida in a Corkscrew Swamp Sanctuary, and it was just like all my reward circuitry was going off seeing that bird. So, so that's absolutely my favorite bird.
That's a right enough answer. But I
What's
think
your
the right
favorite bird?
answer is the cassowary
Ah, the cassowary, yes.
or the emperor penguin. But anyway, those
Those are
facts.
both good choices.
So do we have other questions here? So I'll take one final question because I'm very curious. Do you see any evidence of vertical migration with changes in climate, or is that just not feasible given where the plants are?
Yes, I think that actually I didn't mention that, but vertical migration, meaning going up or down the mountain based on the phenology of the flowers that year, is going to be a huge factor in the hummingbird's resilience in the face of unpredictable changes at our particular field site. So we think that these hummingbirds can move up and down and do move up and down, which should allow them to make these adjustments. Some of our colleagues are actually trying now to put out sound recorders really high up the mountain where nobody wants to go, to see if you can pick up recordings of hummingbird trills to see how high they actually go. So in conversation with my colleagues at Rumble, we sort of share the opinion that vertical migration may be a solution to some of these shifts that we're observing, but we don't yet have great data on that for the broadtails.
Okay. Well, if there are no more questions, what I'd like everybody to do is thank Cassie again. And I want to thank all of you here and everybody on Zoom for making it through our first return to Princeton year of seminars. Keep your eye out because we're going to have a whole new round next year, and I am hopeful that it will be back as normal as possible. Thank you.
Thanks, Gabe.